I'm Kathy Matthews, a neurologist from the University of Iowa, and what I'm going to do with this session is I'm going to introduce Dr. Volker Straub, then I will get back up for 2 seconds, and then I'll introduce our second speaker, Meredith James. So Dr. Straub is a professor of neuromuscular genetics at Newcastle University in the United Kingdom. He has a number of other very long names that I'm going to, in the interest of time, skip, but he is a very accomplished man. His interest in muscle disease is in translational research, and he's made major contributions across neuromuscular diseases to the advancement of clinical research, clinical and translational research. The overall goal of his research center in Newcastle is to accelerate the development and delivery of treatments for patients with neuromuscular disease, and his current research involves application of muscle imaging, use of zebrafish in mouse models, next-generation sequencing, and other omics for the characterization of primary neuromuscular diseases. Dr. Straub.
Thank you very much, Kathy. Thank you very much also for the organizers for inviting me. So Carol Abraham and Catherine Bryant and also Brett Williams. I really appreciate to be here. I think this conference is really a milestone in the collaborative effort to bring improved care and treatment to patients with limb girdle muscular dystrophy. And as you can tell from the session title and also from the title of my talk, it is very much about patient involvement. And I think the fact that this conference has been organized actually by patients shows how much patients are involved. Now, Kathy Matthews said that I work in the United Kingdom, and some of you might wonder what a strange British accent that is. Actually, originally I'm from Germany, but indeed I do live in the United Kingdom. I'm the director of the John Walton Muscular Dystrophy Research Center, and you have heard the name John Walton this morning already because he was the clinician who introduced the term limb girdle muscular dystrophy to the world. So it's actually great to work on what he has started to establish and to come from Newcastle to this wonderful city of Chicago. And so it's really about sharing our knowledge. Knowledge is power, and it's all about empowering patients to be involved in research activities. I think it's really important that patients and families see themselves as a player in progressing the field. Now, this is my disclosure statement. I do work quite a lot with industry, and again, I think that's very important. Within the field of limb girdle muscular dystrophies, I work on diagnostics with industry, and I hope also more and more on clinical trials. I don't think that any of these partnerships and collaboration poses a conflict of interest for my talk. So, very brief overview. I'll talk about trial readiness, and I will very briefly touch on nomenclature because I had some responsibility in changing it, and then, what is a natural history study? And when we talk more about natural history studies, and it was actually great to hear from Sarah Emmons already an introduction about how important natural history studies are and that they have nothing to do with dinosaurs, then Kathy and Meredith will actually look at some more specific trials for collecting natural history data. And in the previous session, you had also heard from Raymond Hummel about the paradigm of clinical trials, and it all starts with a bright idea, and then you have to do basic research, also often referred to as preclinical research, and then hopefully, and that's I guess what you want to see, we move into clinical trials which show that there are effective drugs that will then be accessible to patients. Because the most important thing when we talk about translational research is access to treatments, ultimately. Now, moving into clinical trials, particularly in a field of rare diseases, is a lot more complicated than most people think. And you have already, I think, figured that when it comes to clinical trials, we work in a highly regulated environment. And that makes it often so boring to listen to as patients and families, because you want to know, well, why don't things not move on. We hear about gene therapy now for years, but where is it? Why does it take so long? And I think in summary, it takes so long because states want to make sure that the drugs that you will hopefully get are as safe as possible and as effective as possible as well. And I think that's something that we can all agree on, that of course we want to have drugs that are safe and effective. So in the clinical trials, you test whether a drug is safe and works. But what does trial readiness really mean? Now, you have already heard from Sarah that there is this big international network, TreatMD, a neuromuscular network that really, as a primary concept, wanted to get the neuromuscular field trial ready. And trial readiness means that you have to tackle issues that no individual researcher can really tackle. For example, setting up biobanks, patient registries, guidelines for care, guidelines for diagnosis. So something that you can actually only do together. And together in this case also means together with patient involvement. So all areas where international consensus and collaboration is essential to get things moving. And of course, you want to not always reinvent the wheel, but maximize on existing resources. So this is what the concept of trial readiness is, that industry feels also an incentive. This is the field that is prepared. If we ask them how many patients are there, that they might have a registry and can give you information about this. How do you diagnose the patients? Are there any standards for this? Are there standards of care? Because you can imagine if, let's say, life expectancy for a specific disease is, a 10-year difference in one country to another, you can't have a trial across these countries because it would really jeopardize the trial results. So making sure that there's a level playing field. And I said diagnostic standards is something very important. And you have heard a lot this morning about genetic testing and how important genetic testing is and how complicated it is. And it's partly due to the fact that there are now about 1,000 genetic neuromuscular diseases. And many of them present with weakness in the shoulder girdle muscle, so it's difficult to lift your arms, and with weakness in the pelvic girdle muscle, so it's difficult to walk upstairs or to walk at all. So it's not just the limb girdle muscular dystrophies, but many other diseases that present with these symptoms. And the challenge is to identify now the specific disease. And I'll show you one slide that you have already seen about the problem with the large list of limb girdle muscular dystrophies and how they were originally classified as 1 and 2. And you already heard from Dr. Wicklund this morning that we now have a new classification which looks at the dominant and recessive mode of inheritance. Now, because I initiated this, people also made me responsible that some limb girdle muscular dystrophies were dropped and how this may affect patients. Now, at this meeting, when we decided this was a consensus meeting, there were representatives from patient advocacy group, there were patients there as well, because it's important that if you have a diagnosis, you don't want to change it. But there are also some new forms of limb girdle muscular dystrophies, and we know already that in the next few years, there will be more limb girdle muscular dystrophies. Now, the challenge again is to genetically test them, and one of the projects that we have done in Europe is called MyoSeq. And I'm really grateful that despite the fact that it was a European-based project, many of the funding also came from US patient organizations, and I've listed them there from the LGMD 2D Foundation, the Curtin Peter Foundation, LGMD2I Research Fund, Coalition for Cure Calpain 3, so quite a number of organizations, and the Samantha Brother Foundation as well, that funded this effort, where we looked at 2,000 patients with limb girdle weakness of unknown origin. And just to illustrate again, and you have heard from many of my colleagues that told you about genetic testing and how difficult it is. And I think this library that you see here illustrates the complexity. This is the human genome written down. And all of you have heard that there's a genetic code, and it's actually just 4 letters. And if you would write down the entire genome, so the entirety of all the genes and all the sequences, the DNA information between the genes, for one single human genome, you would fill these 118 big books. And they have numbers, and these numbers refer to the chromosomes. And you can see it's small print, it's 4 letters, there's no punctuation. It's very boring reading. I've started with a Y chromosome, but I think I'm giving it up. Now, these are 3 billion letters, and that's always the difficulty to imagine how many letters are 3 billion letters. That's what 3 billion letters look like. And it might be that one of these letters is different, and it causes limb girdle muscular dystrophy. Now, you could say for a clever computer, it should be fairly easy to find that letter that's different. And actually, that's true. A computer can find this. The real challenge is if the computer finds 3 million different letters, that means 1 in 1,000 of 3 billion. And you need to find, well, which of them is responsible for the muscular dystrophy. That's difficult. And that's why we are struggling to get a diagnosis to everyone. But I don't think that some of the figures that were presented this morning really reflect the full situation. The vast majority of patients with limb girdle muscular dystrophy have a genetic diagnosis, and I think it was really encouraging to see when Dr. Wicklund asked for a list of hands how many patients have a diagnosis, that most of you do have a diagnosis. And we know, and that's similar to what we see in the US, that also in Europe, the most common form is caused by calpain-3 mutations and then by dysferlin or noctumin 5. There are some other genes in green that are not really limb girdle muscular dystrophies, although one could say, and you hear a lot about Duchenne and Becker muscular dystrophy, that the most common form of limb girdle muscular dystrophy is actually Duchenne and Becker muscular dystrophy. That's exactly the problems that they have. Just for historic reasons, they have been assigned to a name of the person who first discovered them. Now, many of the limb girdle muscular dystrophies are extremely rare. And for those patients, it's more frustrating probably because they don't see that in terms of trial readiness and clinical trial programs, a lot of is happening yet. So when it comes to natural history studies, you have to see natural history studies as something in between a patient registry and a clinical trial. So I'm supposed to be an expert in limb girdle muscular dystrophy, but if someone would ask me, can you tell me how much strength a patient with, calpain-3 deficiency would lose over 6 months in his arms? Or how much strength will a dysferlinopathy patient lose in 2 years in terms of the walking distance? And this was something that we couldn't answer. Yeah, we were supposed to be the experts. We know how to diagnose these diseases. We know the patients, of course. But these specific questions about, well, how quickly does the disease decline? How progressive is the disease? These were things where I said, well, that's something that we need to collect. And if you think about the patient registries or clinical trial, it's also a question of investment and robustness of data. So in a patient registry where a patient might initiate the registration and puts in data, it might be a lot less reliable than in a clinical trial where every data entry is monitored independently by someone else, because that's the robustness that is required. And a natural history study is somewhere in between. It's not an intervention, but it does look at progression in a very robust way. And I think the most important message is that the natural history study is really about you. How good are you at a certain point of assessment? That's what we want to assess, and Meredith will talk more about this as a physiotherapist because they play probably the most important role in that assessment. And then it will be interesting to see, well, how is your function and your strength looking 3 months later or 6 months later or 12 months later or 2 years or 5 years later. So we monitor this longitudinal, the change longitudinally. And of course, it would then be really interesting to see, well, how would a patient do if we now introduce a treatment? Can this natural history, the decline, the progression of the disease, that's what's so frustrating about muscular dystrophy, can that be either improved or stopped or slowed down? And we want to use these natural history studies to also look how good are we in assessing a patient. And ideally, if we see, well, the patient tells me he's getting weaker, but I can't measure it, then to improve our measurements. So this is what the natural history study is about. And within the limb girdle muscular dystrophy field, there have been a number of natural history studies. And I haven't listed them all, but we will talk briefly about some that have been done and will also be done in more detail for limb girdle muscular dystrophy 2I or R9, FKRP related. Then Meredith will really cover the dysphilonopathy study, which is the biggest natural history study in limb girdle muscular dystrophies worldwide. There have been already studies in calpainopathy, but new studies I think need to be done now as well, and for the sarcoglycanopathies. And you have heard that the GRASP consortium here is very keen to collect more data in the future. So I'll just show you, it's really a bit of giving you an idea why we do this, how we do it, and what the benefit is from these natural history studies. So this is a 6-year multi-center follow-up study in patients with LGMD2I or R9. There were 4 centers, Newcastle, London, Copenhagen, and Paris. They all had the same genetic change, and we did a comprehensive assessment, including a muscle MRI, and it took about 5 hours, so it's quite burdensome for a patient as well. And what we can do with an MRI is we can actually quantify the amount of fat, and these are the percentages that you read per muscle. And in brackets, you see what a normal person would have. So you can see that a muscular dystrophy patient has a lot more fat in his muscles. And so this is the thigh and the lower leg muscle here. And so we can use MRI and we can use these assessments that I have listed here for breathing, for muscle strength, for muscle function. And we have assessed the patients at a baseline assessment at 6 months and at 12 months. And what was very interesting to us is that the patients tell you, and as a clinician I'm convinced it's true, that over this period of 12 months they are getting weaker. But when the physiotherapist assessed the patient, they were not able, with any of their tests, to demonstrate that the patient indeed got weaker. Only when we did the respiratory function, the breathing, and the muscle MRI, we could see that indeed the patient got weaker. But it was not, we didn't have the tools to assess this really. And this is an example from a patient who's very mildly affected. So the graphs that you see are at baseline in blue and then green in 6 months and red in 12 months. And we did it muscle strength and 6-minute walking distance and the North Star ambulatory assessment. And the score was almost the same over a year. And then we looked at the muscle MRI and we could see that some muscles had almost 100% more fat after a year. So clearly there was progression of the disease, but only the imaging captured this. This doesn't mean that you now not all need to have an MRI. This is part of the study, okay? But it shows you that we can maybe use this to then assess treatments. So if you can stop that muscle is turned into fat with the treatment, then you have a good outcome measure. And then we looked at patients after 6 years as well. The average age was 47.5 years. Patients had, of course, progressed. Not everyone was ambulant anymore. Some now had also ventilation with a respirator, and some patients had heart involvement. But what we saw is that all the muscles, and these are muscle names that are listed, that all the muscles showed an increase in fat. And this is basically what muscular dystrophy means. It's a muscle wasting disease where muscle is turned into fat. That's what we have to stop. And when we looked at all the physical function tests, it was astonishing to see that with some of these, even after 6 years, we couldn't really show progression. And you may have heard about this famous 6-minute walk test. Now, the red lines are those from patients that declined over the 6 years. But there were a few patients that got better. Now, is this that the patient really got better, or is the test just not very good to show progression? So for some individual patients, the test doesn't really reflect progression. But if you lump them all together, then we could actually see that statistically the 6-minute walking test is still applicable. But many of you will know that actually stepping up a stair or onto a box is a real problem for limb girdle muscular dystrophy patients. Rising from a chair can be a real problem, and we saw that that is a lot more meaningful. And I remember patients who told me, look, doctor, once I'm standing, I can walk. The problem is getting up. So, but that's important because that's what we need to learn from you. This is where patient involvement is so important. And the FDA and also the European Medicines Agency in Europe are now listening a lot more careful to what patients feel is actually relevant to them. So this is where we need to work together. So really important information that we get from these natural history studies. And the more information we have, the more trial ready we are to then test an intervention. So thank you very much for your attention. And I'll hand over to Kathy.
Thank you, Volker. That's wonderful. So many of you have participated in the Iowa Wellstone Natural History Study, which is also a natural history study focusing on dystrophic lycanopathies. So the dystrophic lycanopathies are a subset of diseases that are a combination of limb girdle muscular dystrophy and congenital muscular dystrophies. And the overall goal of our center is to improve treatment for patients now and in the future. The dystrophic lycanopathy cohort has been being collected for about 15 years. It represents most of the states in the Union, plus a few other countries. And I just put a couple, we have a number of genes involved. I put the ones that are potentially of most interest to this group. We have 84 patients who've been seen at least once with FKRP mutations and 11 with GMPPB mutations, and many of those patients have limb girdle muscular dystrophy. We also do assessments very similar to the ones that Volker just talked about. Ours are annual, not every 6 months. And I just wanted to point out a couple things. We have 68 subjects now who have been have had more than one evaluation at greater than 6 years of age. They range in age from 6 to 72 years old. And the number of evaluations is from 2 to 14. And this is similar to, this is individual patients, and this shows you the variability in real human data. If you can actually see the little lines, you can see that individual people go up and down. This is the adult population for how quickly people walk 10 meters. And I put this in just to show you part of the complexity of clinical trials. So this is the pediatric data for the same test. And you can see that there's a subset of patients in the pediatric group who really do not change over a number of evaluations, while another subset do decline. So the more we understand about different subsets, the better we are able to design a trial that is going to detect change. So we've used the information, we've shared it with pharmaceutical companies, some of whom are here today to help design trials, and then we've also looked at data to try to help improve diagnosis and care of patients. So if any of you do have a dystrophic lycanopathy related to limb girdle muscular dystrophy, we're happy to talk to you. We're on clinicaltrials.com, or you can contact me. And now it is my pleasure to introduce Meredith James, who is a specialist neuromuscular physical therapist at the John Walton Center for Muscular Dystrophy Research at Newcastle, where she works with Volker. And she is also involved in the clinical and research activities for children and adults with neuromuscular diseases. She apparently lived in Australia previously, and the most interesting thing on her little bio is that her early work included going with the Royal Flying Doctors to the outback of Australia, which I would love to hear more about. So she is going to be talking to you today about the natural history study in dysferlinopathies.
Thanks, Kathy. I'd also like to thank the meeting organisers for the invitation to speak today, and also to the Jane Foundation, who not only support the clinical outcome study for dysferlinopathy that I'm going to speak about, but also support me by contributing to my PhD fees. As Kathy said, I'm an Australian, and I work in Newcastle, which is why I don't have a British accent, much like Volker. Our team is quite global. Ooh. Try for slides. Okay, and my disclosure is that I do a lot of training of physical therapists around the globe in performing outcome measures or physiotherapy tests in natural history and clinical trials. So just a little bit of a background, you'll hear this word outcome measures bandied about a hell of a lot, and what does it actually mean? For me, as a physiotherapist, it's the tests I use, it's the things I make you do when you come and see me, that I try and get data that describes to me how your condition's affecting you, and using measures that are actually appropriate for your condition, that show me and reflect the difficulties that you have, and measures that I can use over time that are actually going to be sensitive to change. So you've seen the slides from Kathy and Volker, where there's a lot of what we would say a lot of noise in the data. We want to see which outcome measures or tests are the best tests that we can use that show how your disease may affect you and how it may change over time, so that when a drug trial comes, we can say to pharmaceutical companies, well, this is the most appropriate outcome measure or physio test to use for this condition at this stage of the disease. The FDA and the European Medicines Agency want us to make sure that our tests have clinical relevance. And so what that means is, if you have difficulty getting out of a chair, which many people do, we need a test that actually reflects that as well. The tests that we do as physiotherapists need to be clinically meaningful. And they do need to be robust, and they need to be measurable, and they need to show change over time. And our outcome measures, they're not just exclusive to physiotherapists, although if you are involved in a natural history study, it's us that you tend to come and see, and we put you through a hell of a lot of assessments. If there's any COS patients in the room, you will know that we take you to the physical therapy room and keep you there for a few hours and put you through a lot of things. Outcome measures can also be things such as the MRI, so the fat fraction that Volker was measuring, and they can be also more and more things, biomarkers, blood tests, things that we get from those types of tests too. I'm going to take us through the COS study. So COS is the clinical outcome study for dysferlinopathy. So it was a study for 2B, or R2 with the new nomenclature. And it was our largest natural history study for limb girdle muscular dystrophy ever, that's ever been in the literature. We were international. So you can see on the map there, we had 5 sites in the US. We had a site in Tokyo, Japan. We had a site in Sydney, Australia. Sadly, I couldn't go and do all those assessments. That's my hometown. We were in Newcastle, we were in Spain, we were in Italy, and we were in Germany. And our patients, we had nearly 200 across the globe, which is a phenomenal amount for a disease that's so rare. And our patients gave up their time to come 6 times over 3 years to come back and get tortured by the physical therapists and have other tests as well. The sort of things that the patients needed to do was strength measurements. We looked at lung function. We looked at time tests. How do you go getting off the floor? How do you go getting up the stairs? We did use the 6-minute walk test because it's something that's quite common in pharmaceutical drug trials in other neuromuscular conditions. And then we use scales of motor performance, and that means things like looking at how you stand, how you walk, how you get out of the chair, how you get off the floor. So we did a kitchen sink of assessments originally. And the reason we did so many tests to start with was that there was not a dysferlin-specific test in the literature. So we needed to start with everything. There'd been small numbers of patients reported, but not consistently over time using the same tests. So the clinical experts came together. We decided to try a lot of things. The Jane Foundation were phenomenally supportive in allowing us to try lots of things first so we could make a refined assessment to have something that was trial ready. So we had the right group of assessments at the end of the study. We picked our muscle tests on the basis of the pattern of weakness that people experience, and then what we were allowed to do is after a year, we looked at all the data from everybody and we were able to make changes. We were able to get rid of the tests that were not specific for dysferlinopathy, and we were able to add in new tests that were going to give us more information, particularly about the hand, which hasn't been well documented in the literature before. And what we can see is in this study, actually, we show small but significant change year on year across all these different tests that we do. So whether we're measuring your strength or your function or your respiratory function, as a group, year on year, there is a small decline. Our most interesting and useful or sensitive tests were our North Star assessment, where we took a modification of the North Star that's used in Duchenne muscular dystrophy, our 10-metre run, and the timed up and go, which is a test where you have to stand up out of a chair, walk 3 metres and sit back down again, and we take the time for that. What a natural history study lets us do, as Volker and Kathy have already explained, is allows us to try and understand how your disease changes. When you come and see me in the clinic, I don't have a crystal ball, and nor do I have the scientific literature yet to actually be able to say, okay, this is what you can expect exactly to happen in the future. However, now that we've done the 3 years and up to 6 years in some sites, we can understand how the disease is changing in everybody, and then we can get into the nitty-gritty of what might be the indicators that tell us how the disease is going to affect you. Specifically, what we've done is we've made a scale of motor performance that is for limb girdle muscular dystrophy. And this scale, we brought it together by trying 2 different scales at the beginning of the study, and then after 1 year, seeing which items were actually relevant for dysferlin and which ones were not useful. This graph that's on the top left with the pink rectangles and the blue rectangles, this is a form of statistics that we use to match people with the items that we ask you to do. So pink is everybody, this is the COS population, and on the left is the weakest patient, and on the right is the strongest patient, and blue is the items of the scale. And what you want to see is a mirroring. You want something that there is going to be appropriate for testing everybody, and that scale does that particularly nicely, except on the right-hand side where we have a group of patients who have got no disease symptoms yet, so they're almost too strong to be captured. The bottom graph is just showing us how people change over time. So your total score is the y-axis, the vertical axis, and the horizontal axis is 3 years of change. And what you can see is that there's just slow change in most people. The blue are the mildest cohort, the red are the moderately affected, and the green are the severe and non-ambulant patients. And what you can see is everyone has a slow deterioration, but within that there are patients having a really rapid decline in their disease progression. And we can see it, and now we need to understand why and who those people are and why that's happened. This is also important for a clinical trial, because when pharmaceutical companies want to run a trial, they want to try and group patients who are very similar in their ability, so that then if the drug is having a good effect, you can capture the drug by using the right tests. And that's what we've been doing. For the COS patients who are in the room, particularly the Columbus cohort, who had to have 3 sorts of strength testing, we needed to compare the manual muscle testing, where we push on you, versus the handheld, where we have the little device that we push, versus the system where you get strapped to the wall with a series of pulleys. Quite time-consuming and quite hard work. We compared all 3 types. They all can show change, but actually our handheld system, where we use the little dynamometer, is probably the most sensitive and useful, and actually it's more time efficient for everybody, because you only need a small piece of equipment and we don't have to strap you to the wall to assess it, which is nice for everybody. We need to understand how a change in your strength actually affects a change in your function and what the relationship is between those two things. So kindly, my colleagues from Nationwide in Columbus have just plotted how your change in strength over 1 year on the vertical axis is versus your change in function on our North Star assessment. And what we can see through that is if you had a greater decline in your strength over the year, you also had a greater decline in your function. And whilst that might seem like common sense, we actually need to document it scientifically to be able to show that as well. We hadn't, in the literature, documented and objectively tested how the upper limb was affected in dysferlinopathy nor other limb girdle muscular dystrophies particularly well. So what we did was we adopted one of our assessments that we made for Duchenne muscular dystrophy a few years back and started using that in the COS trial. So the Jane Foundation was so helpful. We introduced this at year 2 because we realised that we really weren't capturing the difficulties that people were having with their hands. And so we followed the same process. We asked everybody to do this test, and what it does is we look at how well your arms go above your head, can you get a cup to your mouth, and then there's some funny items that involve stacking cans of creamed corn or tomato soup, depending on where you are in the world, pushing on a light, picking up some coins. So they're functional tasks. They're supposed to have clinical relevance. Can you hang your coat up on a hook is like the can item. So each item comes back to something in daily life. We did the same sort of statistics on this test after everybody had done it for a year to see was this going to be a robust and sensitive measure for dysferlinopathy. And it turns out it is. It's really useful and we will continue to use it. These graphs on the right are this type of statistics that I've done, which is called a rash analysis. But again, we're just seeing, do our people and our items, do they marry up well? Or are we asking people to do items that are way too difficult or way too easy, which it would be a waste of everybody's time? And instead, with the performance of upper limb, we've got this really good mirroring again between people's abilities and the tasks that we're asking them to do, except for our very, very strongest patients. You've heard about the patient report outcome measures and how important they are to have patient involvement in this process. We use a scale called the active limb, which essentially is a list of items of daily living, and you just classify whether you find that task easy, difficult, or impossible. How your disease is affecting you in daily life is really important that me as a physio can capture it with the assessments that I'm using as well, and when I'm asking you to do functional things too. So we looked at how the North Star related to the patient report outcome measure. And what this graph is showing us is that actually, if you score higher on the active limb, so you say lots of things are easy, I can get up the stairs, I can walk for a kilometre, I can hang up my coat, I can get off the toilet, it's not having too much difficulty, you also tend to score highly on the North Star assessment. And if you have a lot of tasks that are impossible to do, you then score lower on the North Star. And again, it may seem like a logical answer, but we needed to confirm it with hard data, and we've done that here. So this is a really nice patient report outcome measure. And that's important for informing the pharmaceuticals companies who need to know which assessments to use in their clinical trials, that's a good one to use. Volker's given us a bit of a background about using MRI as an outcome measure, and the particular outcome measure we use with MRI at the moment is fat fraction. So when we look at this muscle, this is a thigh segment, the bright white in the middle is the bone marrow with the black around it being the bone, and then you've got your muscles, and you can see the muscles are different coloured. Some are really dark and some are more white. The dark muscles are the stronger, less affected muscles, and the muscles that have got the white around them or inside them are the muscles that have a higher fatty infiltration. And that's given a score between 0 and 100. And the way that's done is that someone will sit with each of these slices of your legs that are taken in the MRI scanner, and they will draw around the muscles, and then the computer program will calculate how much fat has changed in your muscle, and then we can use that over time to see change like we did in the 2i study. So our next steps for the COS study is to validate all the outcome measures that we investigated during COS1. So A huge thanks to the Jane Foundation again and our patients for undertaking this journey with us again to have a set of trial-ready outcome measures ready to go for clinical trials. We welcome 2 new sites in the USA. If you're not in the COS study already and you have dysphelinopathy, please have a chat and you're interested in participating in this study, and I haven't put you off by saying all the things that we put patients through. Come and see us. We'd be very happy to direct you to your nearest site that's going to be involved. We're also, I know we have an international group here today of patients, so we also have a site in Newcastle. We're in Italy. We have a new site in Denmark and a new site in Chile. So we are a global coverage. We have, for the COS patients that are in the room that aren't aware, we've made it a little bit easier this time. You're not going to be doing as many things because we've been able to refine and tunnel how much stuff we ask you to do. So there's less physiotherapy assessments. We're just using the most sensitive ones, but we have some changes within the cardiac assessment. The MRI, we're really focused on looking at the fat fraction and also looking at biomarkers and how they relate to the disease. Something that was really important for me for this next part of COS is the care discussion. We don't have a set of published standards of care for limb girdle yet, but we need to. We need to get these sorted, and we need to have them. We have them for SMA, we have them for Duchenne, and so what we wanted to do was capture your experience with the disease. How is it affecting you on a daily basis? How does it affect employment? What's your home situation like? And what sort of access to care do you have? So part of the COS2 study is this discussion and a questionnaire at the end to capture that information so that we can start building up an idea globally of what experiences people have to work towards a standards of care document. So that's sort of the items we go through. Falls, do you use splints on your legs? Wheelchairs, are you using one? Are you able to access one? Work, home adaptations. And if anybody would like to come to Vivanti, Jones and I are doing a talk tomorrow on physical therapy, OT and physical medicine, and looking at some of the practical assistance that's available for aids in the home and adaptations and physical therapy. We have to publish and share our knowledge across the scientific community. So we've published several papers from the COS baseline study, and there are more coming out as well, and we shall continue to do so. So the COS study shows that although we made our patient group do lots and lots and lots of things, it was incredibly useful because we actually came up with a package of assessments that we can utilise for clinical trials. We know which assessments are going to be useful for dysphelinopathy, and we can extrapolate that to other limb girdle types. Our patients were phenomenal in their commitment to coming back to us year on year on year and giving up their time and difficulties in staying in local hotels without necessarily the best wheelchair-adapted bathrooms or beds, or trying to get a hoist under the bed. But everybody came back, which was phenomenal. The dropout can be an issue in some trials, and in COS, we just didn't have that problem. It was amazing. We've made, through that trial, our first limb girdle muscular dystrophy specific outcome measure. So that's a real achievement, that's ready to go. And it would not have been possible without our patient cohort. The other thing I'd like to mention, that's our team. Our COS team is the picture on the left, and that's our physiotherapy team on the right in what looks like a sunny day in Newcastle, which is unusual. We've had a photograph taken outside. I'd like to touch on the natural history studies that are going on at Nationwide at the moment. You might have seen, we've already seen several patients. We're just across the hallway here in the San Francisco room. And if you don't have dysphelinopathy and haven't been in COS but would like to be involved in natural history, we are there all weekend till tomorrow about 2. We're there tonight till 8, capturing information from patients by using some physical therapy tests. So the team at Columbus have been collecting natural history data for a couple of years now. They tend to do it in Columbus, but they also travel out to communities in Indiana, and of course here in the San Francisco room, just across the hallway there, just out and turn right, you'll see us, there's a sign. They like to see their patients every 6 months, and they use generally the same sort of tests that I've described and Volker and Catherine have described earlier. So it's quite a few. We're not making everybody do all of those things, but I'm going to talk through a couple of the really interesting novel outcome measures. The 6-minute walk test hasn't necessarily been the best test for limb girdle muscular dystrophy to demonstrate change. So the team at Nationwide, the physiotherapy team there, took the gauntlet and they started doing the 100-metre time test. The course is the same distance as the 6 minutes, so it's 25 metres between 2 cones. But if you can run, you do a run, 4 laps. And if you can walk, then you walk the 4 laps. And then we take the time and we look at the lap times. This is allowing us to delineate between the different types of limb girdles, and it's showing change over time. So this is a really nice test. We are doing it here upstairs as well as part of the natural history data collection this weekend. So that's been lovely that we have a test of walking that is going to be better than the 6-minute walk test. The other thing we needed, simultaneously while we were starting the performance of upper limb assessment for the arm in COS, the Columbus physio team developed the active seated. And this is using the Microsoft Kinect, which has a camera that captures you. You sit down and you can choose whether you like to collect gems, reaching with your arms, and whether you can reach here and use your trunk, or you're reaching here. It captures you whatever your arm ability is, and it captures your trunk ability, which is really important because we don't have any good assessments for the trunk, and this is it. So they've also been very busy capturing lots of information about that. And the really nice thing about the active is, one, it's fun. You can collect gems or squish spider guts across the screen, whatever you prefer. But what it will show is that for anybody who has an ability to lift their arms up like this or like this, whether you're ambulant or non-ambulant, even if your arm function's the same, it captures a difference between the two because of how your trunk is affected. So that's really, really important, and it shows longitudinal change over time. And that's some of the lovely active data, and we would be happy to add lots more data points to that if you want to come and see us over there today or tomorrow. The other thing that's very busy in limb girdle at the moment is activity monitoring. So the Columbus team have a Fitbit 2 study for children with limb girdle muscular dystrophy. The Fitbit is great because it's inexpensive. You can get it wet, so if you wear it in the shower, there's not a sudden scream that, oh, take your Fitbit off. And the children are enrolled in this study, and all the incentives on the Fitbit are turned off. We're just interested in blind collecting everyday activity data. So if anybody has a child that might be interested in participating in that study, come and see the Columbus team in the San Francisco room. So that's thank you to the Columbus team. The last thing we've got, which is also in the San Francisco room and in COS 2 in Newcastle and Columbus, is gait analysis. And this is how we look at your walking. We routinely take your time that you walk 10 metres, but this gives us lots more in-depth information. It allows us to see how you're stepping on the ground, how wide your steps are, how long you spend on each leg, because that can really tell us about how your balance is if you have more time on one leg, as well as your gait velocity. And it shows us the difference between walking with your splints on and walking without, or walking with shoes and walking without. And so that's being collected as well this weekend. So if you are interested, in case I haven't told you enough times, we're there. There's a trial information, not just for this weekend, but if you are interested in the GRASP Consortium or the Fitbit study or the Nationwide Natural History Studies, that's the email address you will need to contact them. Okay, thank you very much.